Good afternoon, ladies and gentlemen, and welcome to the next lecture in the series, Challenges in Forest Ecology. Speaker today is Richard Norby. As usual, a speaker that probably needs very little introduction, but all the same, I will give one. Richard received a PhD in 1981 at the University of Wisconsin. And he's pretty much worked all his career at Oak Ridge National Laboratory. I'm sure many of you are aware of where that is. And he actually retired in 2020, whereby retiring being a very relative term. He's now honorary professor at Birmingham University, as well as a research professor at the University of Tennessee. Richard's work has been pretty much about investigating ecosystem responses to atmospheric and climatic change. He is, or has been, and is still a leader in the responses of forests to increasing atmospheric CO2 levels. So we're going to hear something about these free air carbon enrichment experiments, and he probably is the face of CO2, if you pardon the pun. He's also well known for his work on for New Phytologist, one of the premier journals publishing material on plant and mycorrhizal sciences. He's still an emeritus editor and a trustee. He has a career publishing record and an H index of 105, which is truly remarkable. His work's been cited over 40,000 times, and he has 5 publications which have been cited over 1,000 times. Some of them are the classics of CO2 research in forests. He's going to talk about Amazon face experiment. Is about to start, hypothesis generated from previous face experiments. And Rich, over to you.
Okay, very good. Let me get my share screen up. All right, well, thank you, Douglas, very much for inviting me to present to your group here. I really welcome the opportunity to share information about the free air CO2 enrichment or FACE experiment in the Amazon. Many of you maybe have been hearing about this experiment and wondering, has it started yet? When's it going to start? I've been wondering the same, but I'm really pleased to announce that it will absolutely, positively be starting in this year in June, probably is when we'll start the CO2 treatments. So what I want to do today is describe the experiment to you and a little bit of the background about it and some of the pre-treatment measurements we've made. I also want to talk about some of the synthesis work we've done with previous FACE experiments that then lead to hypotheses that we can test in this new FACE experiment. But I want to start with just a little bit of historical background of the events that led up to this experiment. I think it's always useful to see that sort of development in the history of science. I think an appropriate place to start any kind of work on plant responses to CO2 is with, or at least in the global context, is with the beginning of the Mauna Loa CO2 record by David Keeling, going back to 1958. This graph on the left is the first one I'm aware of showing any of the results. Pretty rough, but what's remarkable about it is Keeling recognized right from the start that seasonal variation is a record of the metabolism of the terrestrial biosphere and therefore a critical part of the global carbon cycle. The more familiar early graph is the one on the right, published in 73, which ended up with a CO2 concentration of about 324. And all right, CO2 now is 100 ppm greater than that first slide, and we all know that. Well, in 73, Keeling and Bakestow published one of the early global modeling efforts, and they introduced in this the biota growth factor, or a beta factor, calling it an adjustable parameter to reflect the degree of CO2 fertilization. This was just a parameter to balance the budget that was driven by atmosphere and ocean aspects. It shows the relative increase in net primary productivity in relation to the log increase in CO2 concentration. Now, as they say in their paper, they're assuming that the land biota responds to gaseous CO2 as do individual plants grown in glasshouses with light, water, nutrients added, because that's the only data on plant responses that was available at the time. They pointed out that if that assumption were true, the land biota would nearly double by 2070. Well, they fully recognize it was a completely unrealistic assumption, and that probably plant growth would not be able to keep pace with fossil fuel consumption. Well, there was a number of experiments that happened then with small plants mostly, and mostly crop plants and so on. Paul Kramer in 1981 published what I think is an especially important paper, because he emphasized the importance of forest responses, which were not being addressed by the current studies on crop plants. You see this graph here is short-term, 20-day, very high, thousands of ppm CO2, not really relevant to global carbon cycle issues. He pointed out that in nature, the rate of photosynthesis and biomass production is more often limited by water and nitrogen deficiency than by low CO2, and that therefore increasing CO2 concentration will probably have little effect if the use of photosynthate is limited by lack of nitrogen. He called for a large coordinated effort to address the role of forests in stabilizing the CO2 concentration. I think this is an especially important paper for shifting the interest in plant response to CO2 to this global context. And it was especially important to me because this paper came out just as I was starting my postdoc in Oak Ridge and getting into these questions. Now, 10 years later, there have been a lot of experiments with tree seedlings in pots and saplings in open-top chambers, as well as a couple of field experiments in low stature systems. And that led Hal Mooney to assemble a group to sort of synthesize what had been the work at the time. But they recognized that the long-term growth responses of woody perennial species, trees, to CO2 enrichment was still speculative because it was all based on these short-term responses, and they don't know whether that could be sustained into a long-term perennial growth. Recognizing also that lab and field experiments were indicating that nutrient availability, water stress, other factors would affect the response, and of course these vary across different systems. So they came up with a hypothetical scheme to predict how different biomes would respond in a relative sense to CO2, indicating that the relative response to CO2 would increase with drought stress and also increase with nutrient availability. But at the time, there's very little, except in the field experiments in the Arctic tussock and in the marsh, Smithsonian marsh. None of these are field experiments. So they called for FACE experiments really should be undertaken in the world's 6 major biomes, as you see here: tundra, boreal forest, temperate forest, tropical forest, grassland, and desert. All of those experiments, one way or the other, have taken place, with the important exception, of course, of the tropical forest. So, 68-year timeline going back to Keeling there has led, with a lot of different experiments of increasing complexity and scale, has been leading to now the Amazon FACE experiment, which we see as really an unprecedented field experiment. There's the website here for the project, lots of really brilliant pictures there and other information, of course. The Amazon FACE experiment, the overarching question would be how will rising CO2 affect the resilience of the forest, the biodiversity it harbors, and the ecosystem services it provides to humanity? It's located in the central Amazon, about 2 degrees south of the equator. It's north of the city of Manaus. And based on some initial LiDAR imagery of tree height, that helped guide us to lay out 6 plots, about 30 meters, 12 years ago now, in the forest. But because of funding disruptions, initially our funding was from the Inter-American Development Bank, but they did not renew the funding because of political instability in Brazil. And only after the UK government got involved through the working with the UK Met Office, were we able to get going again. So tower construction started a couple of years ago to build the face site out. And now the CO2 tanks are in place and vaporizers, control sheds with all of the monitoring for CO2 and then the control for dispersing CO2 into the plenum here. The CO2-enriched air goes into the vent pipes, which you see suspended from the towers surrounding the plots. There's a central walk-up tower for instrumentation for monitoring. And then there's access with a canopy crane for every plot so we can get at the tops of the tall trees. The system was just tested a couple of weeks ago to make sure the CO2, all the valving is working and everything. And here is a very happy site engineer showing the initial record of CO2 going into the plots. So here you see the 6 plots laid out in the forest. Now, you know, one issue that we've had with a lot of face experiments, certainly including the one I worked with in Oak Ridge and at Birmingham and other sites, is fairly small forest sites, so that there's a lot of concern about edge effects. That really is not a concern here. This is amidst a vast, vast forest. Now, one concern, though, is the access road here, typical of many roads in the wet tropics, was muddy and rutted and completely impassable for the heavy construction trucks and eventually the CO2 trucks. So the first step had to be improve that road, which helps us get to the site a lot faster too, which is nice. Give you a little bit of the stand characteristics. It's an old growth, closed canopy, terra firma, or unflooded forest, classified as lowland dense ambrophilous forest. The soil is a highly weathered jarrah ferrosol, or in the US system, an oxisol, heavy clay content, low pH, low concentration of total phosphorus and rock-derived nutrients. Average temperature is about 26.5 degrees, and of course, it's pretty steady across the year. Average rainfall is 2,500 millimeters with a distinct rainy and dry season. There are 6 plots, as I said, about 30 meters in diameter with a buffer area between the towers and the actual measurement plots. 3 of those plots will receive ambient plus 200 ppm CO2. There are about 240 trees per plot. 48 of them are large, greater than 10 centimeter DBH. Canopy height is about 32 meters, LAI about 5.2. One of the really impressive characteristics of the forest, typical of the Amazon forest, and distinct from all of the other face experiments, is the very high biodiversity of this forest. This certainly creates challenges, analytical challenges, samples, how to sample and represent the responses and so on. It also creates some new opportunities, maybe for some creative approaches. You see the total number of plants that have been identified, 423 species, 55 families. And if you just look in the first 2 plots we characterized, just of the large trees, those over 10 centimeters, 52 different species, and only 5 of them are common to both plots. There's 21 families with only 10 common to both plots. So that means replication by species is just not possible. So we have to rely, and also we have to rely on a generic tropical tree allometry to scale up from DBH to biomass, although TLS scans, which we have now, will soon be providing individual tree volumes, which I think will give much more robust estimates of tree biomass. Now, we recognize that models can't capture this functional diversity through a traditional plant functional type approach. I mean, after all these trees, including the lianas and everything, are broadleaf evergreen tropical, despite tremendous differences among them. So the approach we're trying to take is to represent the functional diversity in a trait-based modeling approach. There are six general research areas that the project is designed around: carbon, water, nutrients, of course, which are highly interactive, but different measurements, they interact with each other. This biodiversity issue is sort of overlaid on all of them. And of course, we want to have a strong interaction between the measurements and modeling. I think fairly unique to this project is there is an important socio-environmental component as well of dealing with how high CO2 and climate change can impact the provision of ecosystem services and impact the population of the communities in the Amazon region. There we go. Every kind of lightly replicated experiment like this, it's absolutely essential to have strong pre-treatment measurements. And sometimes there's only maybe a year or less than a year to do that. Because of all of the long lead up to this project, we've had ample opportunity to make many pre-treatment measurements. I'll just talk about a couple of them here. One of them was to scale up the leaf photosynthesis to get a rough estimate of annual GPP, gross primary productivity. This is based on measurements with a Lycor, measured 3 times during the day, once during dry season and in the wet season, and then getting a canopy averaged assimilation using the leaf area distribution, and then scaling the data for a full year using PAR data to exclude periods with low light. I'm having trouble advancing. There we go. Here you see then the assimilation rate is a function of canopy height, and getting an average here, you see a wet season, dry season average, then based on the LAI and the number of hours, get a result of GPP of 4 kilograms of carbon per meter squared per year. Then based on the concurrent measurements of MPP, which I'll mention in a minute, that gives a carbon use efficiency of 0.34. There's other approaches we're looking at, and we want to do this again as we just right before we start the treatments. Another approach is to add autotrophic respiration, which can be pretty difficult to scale up to a ecosystem level, but autotrophic respiration plus MPP. And then also we're installing sun-induced fluorescence, tower-based sun-induced fluorescence monitors. Another great opportunity for pre-treatment data, really important pre-treatment data, I would say, is when the towers were built, they had to excavate down to a meter for the foundations for the towers. So we had an excellent opportunity to sample the soil and the roots down to a meter, actually down to 1.5 meters, in a number of different plots, all surrounding each of the experimental plots. So there's a lot of data both on roots and on soil biogeochemistry and enzymes and elements and so on. Here's some of the root data. Not surprisingly, the density of roots is greatest in the top 5 centimeters and decreasing with depth. If you took the cumulative amount of roots as a function of depth on the right, the data points are the blue dots there. And it's useful to try, and that indicates that 36% of the fine root mass was in the top 5 centimeters, but 75% in the top 30, and only 1% deeper than a meter. But one of the hypotheses we'll be wanting to test is whether as in previous CO2 experiments, whether elevated CO2 drove a deeper root distribution. Now, it's really, the root distribution can be an important input to models, so it's useful to be able to express the data in a modeling context. One approach that's been frequently used in models is the beta fit. Rob Jackson popularized this in particular. That's the green line. You can see it didn't fit all that well, especially in sort of this middle depth. And Rob gave the summary for tropical evergreen biome, which is the dotted line, and that also sort of doesn't do all that great of a job in the dots. But there's another two-factor model from Zhang that was specifically encouraging for the model data interaction. I think net primary productivity is an important factor for evaluating forest responses to elevated CO2. It's been really useful as a benchmark for models and a way to compare different experiments. MPP describes the rate at which the autotrophs in the ecosystem produce net useful chemical energy available for heterotrophs in the food web. We measure it by calculating the annual production of wood using diameter measurements and then allometry to scale up to biomass and biomass production. Leaf production based on leaf and reproductive output based on litter traps underneath. Fine roots based on mini rhizotron measurements. And then there's other components that are not currently being included, but including exudation and volatiles and so on, that we do plan to incorporate as we move forward. The assessment then of MPP is shown here, and in comparison with similar measurements at the ORNL FACE experiment that I was involved in and ongoing at the Birmingham, the BIFOR experiment. Not surprisingly, the MPP is greater in the Amazon, but it's interesting to see how much greater fraction of the MPP is in fine roots in the Amazon than in certainly compared to the Oak Ridge. So the MPP gives a notion of the distribution to leaves, wood, and roots. And that's important because these components have different turnover times, and that carbon turnover affects the biogeochemical processes and the feedbacks between the tree and the environment. Well, as I said, in the first round of CO2 experiments, we really wanted to make sure the FACE experiments were helping to inform models. So those of us involved in these first four FACE experiments that had MPP results got together to synthesize the work, and I won't go into too many details here, but basically plotting the MPP in ambient CO2 versus that in elevated. The points of the different species and the different experiments in different years all fell across a nice line indicating a 23% increase on average response to elevated CO2. And this is across a wide range of MPP, which is pretty impressive, we thought. But we also certainly recognize that this median response was masking spatial and temporal variability, interactions with other global change factors. Warming, for example, could really be important. Nitrogen feedbacks could really limit the response over the longer term. And of course, these are all temperate forests. The analysis did not include tropical or boreal forests. They're all young forests, so the question immediately arose of whether the responses would persist in older forests. Nevertheless, the synthesis has provided a useful benchmark for models, which I was pleased at because that was the intention of this exercise in the first place. But now, with a couple of new experiments, the EUC-FACE experiment in Australia in a eucalyptus forest, and the BIFOR FACE experiment in central England in an old oak woodland, in addition to additional data from the original experiments and recalculated data from them, we're able to expand the inference space and redo this analysis and question, is that 23% response that we reported 20 years ago, is it still valid? Well, the first thing to consider is, especially with the two experiments happening 20 years later, CO2, ambient CO2, of course, has risen a lot since then. And all these experiments differed a little bit in what concentration they were working with. So, and you can see here plotting the responses, relative response to CO2 against the degree, the percentage increase in CO2. This explains 70% of that variation. So we can use this regression to adjust the MPP to the average 41% about here increase in CO2 and put everything on an equal basis. So the subsequent graphs I'll be showing now have this adjusted MPP response. And here showing the graph, the data in the same way as we did 20 years ago, you see they still line up nicely. Now the increase to the 41% increase instead of 23% is 21.8%, not much of a change. This corresponds to a beta factor as Keeling presented it of 0.57. Now Keeling's used in an empirical, in his initial model, a beta of 0.56. So after what, 68 years and I don't know how many millions of dollars and hours of labor, we moved from beta of 0.56 to 0.57. But I think we have a lot more confidence in the data now, so it's not a loss. We are also this time looked at above ground wood biomass increase because there's a lot more data there, including Bangor face experiment that Douglas was involved in. So doing the analysis the same way, we see the wood biomass increased 18%, a little bit less. There's differences in the allocation patterns in these experiments. At Duke University, a face experiment, there was a shift in allocation toward wood. At the Bifor experiment, there's increased wood production, but not in fine roots or leaves. At ORNL, the increase in MPP was primarily in fine roots. Uke face didn't see any increase in wood production. Overall, the wood fraction in these experiments was 58% in ambient CO2, 56% in elevated CO2, so not much of a difference there. Now, as we started up this, what we call second generation of face experiments, we really wanted to continue the being able to inform models. So this time we try to get together with the modeling ahead of the experiments, develop a, sorry, you can't see all those comments, but develop a protocol for the model data interaction and come up with some standard sampling and data protocols, generate questions for a cross-site synthesis. And there was 4 projects we're looking at, but the Swede face never happened. So it's really just the 3. Some of the key issues we considered are stand age and maturity, which did differ across these 3 sites, and all of which are much older sites than the original face experiments we worked with. Nutrient limitations are especially important. And if we look at this map generated by Xiaozhen Yang at Oak Ridge National Lab, based on phosphorus limitation versus nitrogen limitation, all based on soil series. So a fairly large scale analysis. But this would suggest that the Amazon face experiment and the UC face experiment would be in an area of phosphorus limitation, whereas Bifor face is maybe more likely to be nitrogen limited. And of course, the Oak Ridge and Duke face experiments here in the area of more likely nitrogen limitation. Can also consider the temperature, because temperature varies, ambient temperature varies a lot over these 3 sites, as does water. And I mentioned biodiversity, which of course is especially different in the Amazon face experiment. So we can use the synthesis experiment now to generate some relationships based on some of these considerations. So the first graph, you look at the relative response of CO2 to mean annual temperature. Maybe surprisingly, it shows a declining relationship This is the opposite of what was presented, of what was predicted in a number of models, which suggested increasing response with increasing temperature, primarily due to the interaction at the photosynthesis level. I think, however, there's a confounding of effects between temperature and nutrient interactions here. And, we don't have enough data to do a full multi-factor kind of analysis. There's no relationship with mean annual precipitation or various other indices looked at of climate indices related to water. Total nitrogen is almost certainly an important factor in explaining CO2 responses. Progressive nitrogen limitation was documented in the ORNL phase experiment. We think the growth response at the Birmingham Bifor phase experiment is sustained by high nitrogen deposition, but total nitrogen in soil is really not the best metric for evaluating CO2 times nitrogen interactions. And the more other factors, nitrogen availability and so on, are really hard to characterize across sites from exact data. So we don't see too much help here. But with phosphorus, there is a strong relationship of declining response with declining phosphorus availability, although recognize this curve is very much dominated by the Rhinelander phase, which is set up in an old potato field, so high residual phosphorus. Now, there's a lot of predictions that I think what it says there is that older, more mature forest stands would respond less to elevated CO2. And a lot of that was apparently supported by early results from Uke phase and Bifor phase here. But as I pointed out before, when you normalize the results to the CO2 concentration or the degree of CO2 enrichment used, that response goes away. So the prediction based on these data that older stands would have a less response is really not supported. Sorry? Well, now, for what do these predictions mean for the Amazon phase experiment? Well, the stars here represent where on these curves the Amazon is. And of course, the high temperature in the Amazon would suggest very little response to elevated CO2 based on this relationship. And very low phosphorus, also a low response. Stand age is really quite large, but that, based on this analysis, would suggest no difference. One important thing, though, is that the intended enrichment of 200 ppm or 46% over ambient should really aid in the detection of response. Now, there's other analyses that lead to hypotheses for the Amazon phase experiment, and let me run over some of those. First of all, I showed earlier the graph that the scheme that Hal Mooney came up with. This is the same thing, just in a little fancier version of it. But in this scheme, the wet tropical low water stress and low nutrients. Mingkai Zhang, publishing from the Uke phase forests, initially suggested that mature forests won't respond. And that's still a valid hypothesis, although right now I would say that's not supported. Shortly after our 2005 synthesis, Thomas Hickler used the LPJ model to predict global MPP response to CO2. And he compared it with the 23% that we suggested in our synthesis, and pointed out that the global response and the temperate forest response were really fairly close to that degree of enhancement. But he made it emphasize that the boreal forest, being colder, would have likely a smaller response, and tropical forest with a higher temperature, greater response. I should point out that this version of the LPJ had no nutrient interaction in it at all. So this is very much just driven by the photosynthesis response. So more recently, Katrine Fleischer led a multi-model comparison effort, all generated from generated with Amazon Face data, and not response data, but the site characteristics, and showed that the models that had a nitrogen interaction showed a lesser response than the carbon-only models, and those that also had a phosphorus cycle in it had even less of a response, either of GPP or MPP, suggesting that, at least for Amazon Face, the phosphorus limitation would reduce elevated CO2 response. Now, another hypothesis comes from Christian Kerner, who I saw is online here. So he's emphasized that the ultimate response is driven by turnover and not production, and always reminds us that MPP is not carbon sequestration. And that, of course, is absolutely true. So we have to consider of will FACE resolve these questions about elevated CO2 and carbon storage. We have to recognize that the change in forest biomass is equal to new production minus mortality. Well, mortality, especially in a big forest, is very difficult to measure, especially in 30-meter, almost impossible in 30-meter diameter plots. In fact, in our FACE plots, if you just took over 8 plots at the time and over a number of years. Pardon? That most of the trees that died were very small trees, very few trees. The orange, only 3% of the trees that died were very large. But 10 trees accounted for 74% of the lost biomass, but that's spread over a lot of different plots and a number of different years. Basically, the FACE experiments cannot provide relevant data on tree mortality. Now, if we look at this analysis from Brno, which projected a loss in net biomass increment in the tropical forest, that's a net effect of a slight increase in production, but a much larger increase in mortality over the time. Well, I would submit that the Amazon FACE experiment can inform this part of the analysis. But to really get at the essential questions of carbon storage, we have to link this kind of analysis up with remote sensing, regional studies, and modeling that can get at the other larger scale issues. So I want to end it there. Let me, if I can advance. Why can't I advance? There we go. Acknowledge this project. It's coordinated by the IMPA, the National Research Institute for Amazonia, the University of Campinas in Brazil, and the UK Met Office, which manages the funds from the US government, and the other funding agencies of various Brazilian organizations. This whole effort over all these years has been driven very much by Beto Casada and David Lapola, who've coordinated the whole thing. There's some of the leaders you see here for the different components. It's been a great group of many students, postdocs, senior scientists from Brazil, UK, Germany, Austria, Netherlands, Finland, and the US. I've probably forgotten some countries or people, but it's been a lot of fun to work on, and I'm really looking forward to finally seeing the CO2 go on and start getting some relevant measurements. So I will stop there. And I can stop sharing.
Yeah, thank you very much, Richard. So we have time for questions. How we normally do this is we write the questions in the chat box. It seems to be the most efficient system.
Okay.
So you can see the chat box, can't you, Rich? No?
Yes, I do.
Yeah. So I've got the wrong chat box, haven't
I think I do.
I? Yeah. Okay. So if nobody starts off, I will start off first of all, just to get it going. How do you deal with the biodiversity of this forest in terms of what is in the rings? This seems to be a
Yeah.
very, very difficult question.
Well, it is. It is absolutely completely different from the kind of issues I certainly had to deal with before. Um, we just have to get around any notion of replication by species. That's just not possible. So, and it's also really almost, well, for tree growth, we can measure every tree and just get the total for the plot. That's great. But for a lot of other samples, when you're sampling leaves or measuring photosynthesis, we can't measure every tree. So the idea is to try to partition the measurements based on functional types. And there's been a lot of work in trying to define what a functional type is based on wood architecture and some other traits like that. That's still an ongoing process. I'm not convinced necessarily how well it will work, but we have to try something like that. So, of course, plot level measurements should be okay where you're not looking at species anyway. And a lot of the measurements, such as below ground, a lot of the below ground measurements, as long as you sample enough, hopefully you capture that variability. But it's a real problem. But on the other hand, it's an interesting challenge, put it that way.
Yeah, most definitely, most definitely. So, I don't know if anybody has written in there. I just can't see it, but I'll keep it going. What about something very, very close to my heart? How are you going to deal with the roots, and how are you going to deal with this very high root biomass?
Well, first of all, the initial data I showed was from mini rhizotrons, and I think you know all of the concerns about mini rhizotrons. I would still maintain they're the best way to get root productivity despite all of their faults.
Yeah.
Especially difficult in the tropics because of the high clay, they get clouded up, and high water problems. And in fact, we had to reinstall the tubes. We have new mini rhizotrons in now. There's only 5 per plot. Ideally, there'd be 20 per plot, but it's very, very labor intensive. So that's a problem. In between the times we had mini rhizotrons in, we're doing it with we're measuring with ingrowth cores, which I've never quite thought was the best way to measure root productivity, but it's better than nothing. But yeah, I think it's the same problem you get in any system, whether it's diverse or not, is that the variability across the plots is going to be high. And the only way around that is with intensive sampling. And intensive sampling is tough to do with such a labor intensive approach. So I don't have a magic answer to that.
Yeah.
But it is an important, we're not going to ignore it.
I'm sure, absolutely right that all the problems are the problems we always face. It's just a bit more difficult. So I guess you can see the question.
Yeah, okay. Where do you expect an interactive effect of CO2 and changes in biodiversity? Well, there are a lot of people who are interested in that. Of course, the vast amount of productivity is in the big trees, and they're not going to change. Well, one big tree might die. Hopefully, it won't knock down the whole system, which is a possibility, but we won't talk about that. But there could be some changes in the understory, I suppose. They don't, and that might be of interest. There are a lot of people who are very keen on trying to predict that. There was a open time. I didn't mention anything. There was interim while we were waiting for the full funding, smaller scale open top chamber studies in the understory that tried to address some biodiversity issues. But I personally, I like to be, I guess, maybe a bit provocative to some of my colleagues who think that the difference in species responses is going to be a really key issue. I will maintain, I don't know if I believe this or not, but I maintain that basically all C3 plants respond the same. Now, not all C3 species respond the same. Now, of course, the individual, depending on where it is and the nutrient environment, whether it's in the understory or overstory, that affects the response. But that's not a species trait. That's an individual trait. So I say that to be a little bit provocative, and that really upsets people who want to see these key response will be how different species respond differently. No. Christian Kerner asked how many years of stem growth data are available. It goes all the way, it goes back to when we first laid out the plots, which was 2014. The first couple of years were measured a little bit differently, so there's a little problem there, but there's good data for the last few years. And as I said, we've been scaling up based on a generic tropical tree allometry. I really hope very soon to get individual tree volumes from the terrestrial laser scan, and then we'll have a much more robust way to get at it. There's actually older data on a subset of some of the plants because Nira Higuchi was doing some measurements in part of the plots we're measuring. So that some of that data goes way back. It's whether it's comparable data or not, I'm not perfectly sure. Okay, let's see. We have experiments typically elevate CO2 relatively abruptly and maintain at a higher level. True. Yeah, in the real world, it increases gradually. And this has been an issue that's been talked about, I think, from the beginning of FACE experiments, the so-called square wave of the CO2 response. I think one important aspect of analyzing the data is the first year of response really isn't all that relevant, especially like in the Amazon, the leaves will not have been, the leaves are last year's leaves. So they developed under ambient CO2 and now they're in elevated CO2. I think you have to have a longer term perspective on it. The various approaches people have used are trying to ramp up the CO2 though, which they tried in UGFACE, it just creates confusing data and it really doesn't address the question. Sure, it could be considered a limitation of FACE experiments, but I don't think there's a way around it. And we do recognize that it's not a representation of necessarily the future forest. It's describing processes that then hopefully can inform a model.
If nobody's written anything in the chat box, I have another question, which is, are all the plants arbuscular mycorrhizal?
I think so, yeah.
Everybody looked at this? And
yeah. I mean, I know there's some people who've maintained that tropical trees are always that. That's not true.
No.
But as far as I know, they're all arbuscular. There's also somewhat related, although there's some species that are capable of nitrogen fixation, some ingas, they are not nodulated in the forest. In an open-top chamber experiment, interestingly though, with inga seedlings, when they were provided fertilizer phosphorus, they did become nodulated. But in the forest, we haven't seen any nitrogen-fixing nodules.
Okay. The other thing is that are all of the trees evergreens or are they deciduous? Yeah. So in which case do you know?
I mean, they all drop their leaves at different times, but they're all
So do you know what
the forest is never leafless?
Yeah.
Never.
Okay. But do you know what the turnover of leaves are within
Well, yeah, of course it varies with species, but on average, based on measurements made a few years ago, the turnover time is about 1 year, which is convenient. That's an average. There were some very detailed measurements made in the canopy about, I don't know, 6 or 8 years ago. This is the advantage of all this pre-treatment time of, actually tagging green leaves and tracking their phenology. And I think there'll be a lot more work on phenology, both from high resolution photography and other approaches, but on average, the turnover time is about 1 year.
Okay, there's another question.
Okay, this is the question about the environmental impact of the CO2 enrichment in the atmosphere in terms of greenhouse gas emission. That's always been a question that's come up with FACE experiments. For the most part, the answer is there's really very little impact. because like the CO2 I used in the Oak Ridge face experiments and other ones was a byproduct of fertilizer production. So the CO2 was a waste product. It was going to go into the atmosphere at the fertilizer factory. Instead, it's released at our face site. So the only cost really is the transportation of it. So there's really not much impact. It's also, that's not necessarily going to be true, unfortunately, for this experiment in the Amazon. And people have calculated how much CO2 relative to an airplane flight or something. And frankly, I don't remember the answer on that. But so there is some release of fossil CO2 into the atmosphere because of it. Same way there is in most experiments, actually. You're not going to see any direct effects at all distant from the plots. I mean, that dissipates in the atmosphere very, very quickly. So it's only in terms of global CO2 emissions that it's a factor. And I wish we were also getting a non-fossil source, and we are looking at other alternatives right now, but it's always been an issue with face experiments.
Yeah, but the total amount released is not exactly. We're talking about.
No, it's not huge.
Yeah,
but it's not zero either.
No, it's not zero, but I mean it's.
Yeah, and I would consider it irrelevant when it is indeed a waste product, recycled waste product. But
do you know what the consumption is? How many tons a day?
Oh, geez, I don't remember.
Okay, yeah.
We've worked out what the need will be. One of the issues, this is certainly a concern on how well of a treatment we're going to have, is that the wind is very low here. And you need wind to dissipate, to distribute the CO2 into the plots. So at least the initial testing seemed to work out okay, but there'll be more testing over the next month here.
I can contribute to this. We used about 2 tons per day per ring.
Yeah.
And that is food quality CO2. So we buy it from the food market, like if you buy some mineral water or some sealed food, it's the same source. So what we pay is actually the price for cleaning the waste gas.
Yeah.
So instead of leaving it out through the chimney
and transportation of it
from the consumer industry.
Yeah.
And so actually the contribution to the atmosphere is zero, because if we
unfortunately
would go to Coke or to something else.
Yeah, unfortunately that's not necessarily going to be true for the Amazon face.
Okay.
There's a company there that's burning fossil fuels to put bubbles in Coca-Cola.
Yeah.
Which is a little bit disturbing, isn't it?
Yeah.
Yeah, then drink less Coca-Cola, I guess. Okay. So that's about it. We don't seem to be any other questions. Again, Rich, thanks you very much. That's a great talk. Thank you for coming. Thank you for your time. And
happy to have further conversations with anyone. Just send me an email or
Yeah. For all those people listening, then the next seminar is next month. I think it's the 6th of May. Will be Christoph Leuschner from University of Göttingen. He'll be talking about drought and heat resistant trees of Central Europe. And that's it, I guess. I thank you very much again for coming. I noticed in the chat box that there's a number of people I've never seen before. If you wish us to send you the link for the next talks, particularly the University of Neuchâtel, which looks rather interesting, then tell me, send me an email, and I'll make sure that you're on the mailing list. Okay, so nothing else to say. Again, thank you very much, Rich. Great talk.
Thank you.
For those that celebrate it, have a happy Easter. Don't eat too many eggs, and look forward to seeing you next month. Okay? Thank you very much. Bye-bye.
Bye-bye.